Hi, my name is Maedeh Mozneb. I am a PhD candidate working with Professor Chenjong Lee at Florida International University. I'm working on Thrust Area 2 of the CellMed ERC program with the focus of sensors testbed development. Our lab focuses on design and development of several different types of sensors, ranging from optical, electrochemical, and nanosensors to plasmonics and impedance-based ones for different applications. So today I will be going to talk about our plasmonic-based sensors, how the device works, and challenges that we have seen using SPR specifically, and also the applications in the CellMed. How does this tie into CellMed? So the sensors that we have in our lab is typically the optical sensors that we use is fluorescent-based or plasmonic-based sensors. The fluorescent-based sensors include the ATP, oxygen nanosensor, lactate, and glucose. And our plasmonic-based sensors, which is going to be the focus of this video, would be surface plasmon resonance based sensors and also localized ones. Electrochemical sensors are ranging from lateral flow to vertical flow test strips, and glucose and lactate sensors as well. Then we have all sorts of MEMS-based devices and microfluidic input devices, and we also have different designs of microelectrodes ranging from 3-barrel microelectrodes to single ones. So to just talk about what is surface plasmon resonance, surface plasmon resonance is a phenomenon that occurs when polarized light hits a metal film at the interface of media with different refractive indices. SPR techniques excite and detect collective oscillations of free electrons at the surface. They're also known as the surface plasmons. Via the Kretschmann configuration, in which light is focused onto a metal film through a glass prism and the subsequent reflection is detected, which you can technically see in the left figure. So once the light is shine at the specific wavelength, it provides the crucial conditions for the light to have a total internal reflection within the SPR. And once that happens, and when the magnitude of the electromagnetic waves propagating on the surface equals the magnitude of the evanescent wave dissipating away from the surface in a vertical plane perpendicular to the surface of the chip, the resonance of surface plasmons occur. And that evanescent wave within its sensitive range, which is less than 200 nanometers, is highly sensitive to small changes in the mass, such as an antibody-antigen interaction, and that results in a change in refractive index unit, which is read then by the SPR, as you can see in the GIF on the right side, bottom right. And then it is read by SPR in a delta theta in millidegrees change in the reflected light, and that can be translated into how much concentration, kinetic analysis, association, dissociation constants can all be extracted from that information. So why do we even use SPR? So SPR can be used for so many applications for multiple purposes, such as antibody-antigen binding kinetics, molecular interactions kinetics, like association, dissociation constants. It has control over orientation of proteins on the surface with specific modules that the device provides. Electrical stimulation and electrochemical sensing is also possible. Vesicle release analysis from the cells, as our lab has been one of the pioneers in that area. Cell attachment monitoring in real time, and cellular micromotion analysis also in real time can be done using SPR. Now, the study of molecular binding process is a key aspect to many fields of research, ranging from life sciences to environmental safety, determining which molecule interacts, how they interact, and why they interact could be really important in terms of information that is necessary later on for air quality, air water quality, drug testings, higher performance of material, so on. So several technologies exist currently that offer such applications like ELISA, QCM, and ITC, but few of them have the opportunity of surface plasmon resonance that enables high sensitivity, real-time monitoring, label-free detection, kinetic rate constant evaluation, quantitative evaluation, and also low volume sample consumption with the SPR. So also SPR is easy to perform and can be cost-effective solution to future uses. Now, the SPR device that we have in our lab is a BI 2000 from Biosensing Instruments device. The capabilities that it has two channels. It has an injection port for injecting of the analyte or drug molecules. It has two injection channels. It can operate in parallel or series with those two channels. And there are different modules to it. You can see two of them at the bottom of this figure. On the left one, you can see the static module that technically it has a reservoir for media. You can put in media and you mount the chip on top of the prism. Here would be the prism. And then the module goes on top of it. You input the chip on top of the prism here, and then one of these modules are going to be clicked into this going on top of the prism and the chip, and then this injection port over here we can inject drugs through goes all the way through the tubings over here into the device, and then out from the other one, which is the outlet going through the waste. You can select to have either a parallel or a series, in parallel or in serial readout from this device. The static module is going to provide a non-flow situation for your analysis, if you, depending on what kind of a technique you're using, what kind of a design of experiments you have. And the flow-through one has a microfluidic on the other side, which has an injection an injection port that has fluid going through both channels and then out from the other side. It also has a module that can have electrochemical sensing and a flow through at the same time, which technically has three electrochemical sensors input into it. The gold sensor is going to be a working electrode, there's going to be a reference electrode and a counter electrode, as well as you can have drug injections at the same time with the electrochemical one. And the newer models that are out there with biosensing instruments also have microscopy, they have a bright field, they have a chamber for temperature and CO2 control. So how does this all tie into CellMed? How can we tie this into CellMed is, as you can see from the diagram, to develop our required test bed in the most efficient way, we must cover all the fundamental knowledge necessary for the design and development of our technology elements, which will be components later feeding into each test bed. And as a part of that, it is highly essential to understand what is a cellular interaction in terms of their metabolic function, action potential propagation, and force of contraction change within a 3D construct. And to have a thorough information about the 3D construct, it is best not to disturb the tissue and to have a minimally invasive approach for any sort of sensing. SPR is a non-invasive, label-free technique that can help with targets like tissue function monitoring, given that the concept of such application is proven. We also have other types of sensors, such as our oxygen sensor is also optical based, which is non-invasive. It is directly taken up by the cells, so that would also lie into this project that we want to non-invasively monitor the functions of the tissue in real time. So previous work that has been done with our lab is that we have proven for the first time, our lab was one of the leads on developing a cell-integrated platform on SPR with culturing the cells on the microfluidic top gasket and modifying the sensor chip for the release from the cells. Now, our previous work has been developing this module, and the device was later used and tested for SPR response of VEGF of SKOV3 cells released with VEGFR interaction, which is the red line over here you can see. So it's a VEGF-VEGFR interaction. And also the VEGFR interaction under bevasica under a drug-induced situation. So a cell release without the drug with correspondence on the sensor chip, and a cell release with the drug injection under the same condition. So for the first time, we reported direct and real-time measurement of drug effects to the VEGF-VEGFR angiogenic switch on live carcinoma cells. We could have monitored all the release of such a molecule from a cell detected on the SPR and see the signal change due to a drug test. With possibility of cells being integrated into a highly sensitive and label-free system like SPR, it has a lot of potential in measuring several aspects of cardiac tissue as well as vascularized tissue in its platform. Due to the microfluidic base that it has on top, we can also vascularize the constructs of the tissue that we put on top of the cells and connect it with the SPR chip. Now, some of the challenges that might come up in the future studies is technically the optical constructs of SPR is a little bit complicated, and it might take time to construct them in a way that can be applicable in an incubator or be portable design. SPR studies are not temperature or CO2 controlled at this point, and that can have a large effect on the cells, although we can reference out a lot of the disturbances, but still it would be ideal to have a chamber provided on top of SPR that is temperature and CO2 controlled for prolonged studies. The sensors are reusable, but the thin layer of gold, which is 45 nanometers on top of them, can be washed away after some time. A couple of uses can actually damage the gold layer a little bit more, so it might be beneficial to think about the reusability of putting these gold chips within a microfluidic and then have a portable optical device attached to it. The 3D tissue maintenance and fabrication will be challenging. It's not impossible because it's a 2D plane that we're working with, but we can scaffold it. We can integrate the microfluidic on top of it with it, and then have the cells grow in a 3D manner on top of the chip. And also incorporation of a bright field would be very helpful because you can monitor at the same time how the cells are moving, how the cells are interacting, behaving, and then you can also relate it directly to the SPR signal. So this is a surface plasmon resonance device we have, the biosensing instruments BI2000, and it is connected with micro pump, which technically pumps through the fluid all through these channels. We can select all sorts of parameters on it. Technically, you want to have fluid going through with a very high rate, like 150 microliter per minute, because you want the flow to actually go through the channels and cover everything. There's no bubbles left over in all these channels because all those bubbles are going to give signal noises. And then after that 150, which technically runs for 2 minutes, you will dial it back down to whatever your desired flow rate is, which in our case is 10 microliter per minute. So the SPR, as I've mentioned before, it has an injection port over here. It has channel 1, channel 2 selection, which it will say if I want to have the fluid going through channel 1 or is channel 2. And this is the module that will have the fluid flow going through. And it has a microfluidic base that has injection, channel 1, channel 2, and then an output that goes to waste. That will go on top of the chip. So to start your SPR experiments, you would need to have, this is the prism on top of here. If there's a viewing window also, you can see like a black line, like a straight black line on it. First thing that you do is you use an index fluid, which is provided by the company. It has a known refractive index unit, and it is important in calculations to use that. So you take about a little bit of a drop of it, about 2 millimeter in diameter, put it in the center of this prism. So you don't want to put too much because they're gonna float around, so you just put a little bit in there. And then you can take your chip. The gold chips are also provided by the company. You can also make them. We made them ourselves before, and it's just a soft lithography photolithography process, straightforward. You can culture the cells on top of these chips, and so you take that out from your petri dish. Usually we do that in the flow hood so that we don't have any contaminations going through our samples. But here's going to be your sensor chip. It has both sides on it. There's a gold side and there's a chromium side. You can technically see that the gold side is less shiny, more opaque, and then the chromium side is more shiny and with a little bit of a more a reddish color to it. You want to dry it out on the side that goes on top of the index fluid, which is the chromium side. It is always gold face up, chromium face down. So I will real quickly dry this out. Any fluid on that side is going to interfere probably with your the sample reading, so you want to have the least amount. Then you would take a Kim wipe and wipe the other side, tap it on a Kim wipe so that any drops of fluid is dried out from it. You will put one side down, tilt it as if you're mounting a slide on top of the immunostaining slide on top of the microscope slips. So after you have inputted that in there, you would align it, then you would lift up your module, will come down onto this. You will tighten that up. Now your injection is running. You are in a load position, not the injection position, which is on this side. You are on channel 1, and after that you want to have fluid going through, which I will start it right now, and that will have fluid just running through it for 2 minutes. And I will look through the window over here to see a break in that straight line. So the black straight line should be in 2 pieces, and then that 2 pieces are saying that you can read both from these 2 channels and there's no leak in between. Okay. And just for demonstration, I wanted to show how the fluid is moving through these channels. I'm not sure if it's going to be visible, but we can try. I'm going to be injecting 100 microliter of a buffer solution through the injection port, which is with the syringe that is also provided with the company. So we're going to have 100 microliters taken out. Again, you want to not have bubbles in your system as much as possible to delete any interferences. So for injection, you will put the syringe through this knob over here, completely in, and you want to fill it up with 100 microliter, 100, 120, because the sample loop is 100 microliter, and you want to do a little bit more than that to fill it completely. So I will inject very slowly, because otherwise it will leak. And the reason for this is that we want to track where the fluid goes. Without taking it out, you will switch it. This is in load position right now. You will switch it into injection position, and it's on channel 1 right now, parallel. So it will go through both lines. So you can see if you can track the fluid coming out from here. Hopefully it should be turning red, more or less. This is going to be the interface for the biosensing instruments. We have flow through. This is the saved data from before, and this one is an active injection right now on a bare gold chip. So you have an injection port, and when you turn this back on, it goes on to loading, and then when you inject it, it goes on to injection. You can control how long the time is for the injection. You can change the parameters of your plots, getting the maximum data out of them. You can change from flow injection to electrochemistry mode, and then all data analysis from there goes to the BIDA, which is the kinetic analysis. It automatically puts a configuration on top of your selected areas, and then it can directly calculate the association dissociation constants for it.